So good morning, good afternoon, everyone. Um, so as usual, this meeting will be recorded. And then if anything occur that you want us to edit, please reach out before the end of the meeting. So our agenda for today is, so in the last few weeks, we've talked a lot about the NeuroPixel analysis. And last week, we talked about sort of looking into behavior, but I reached out to a couple of collaborators on the project to try to organize a session around Mesoscope data validation. And some of these are here today. A few of, I think, are new students, but Farzaneh Najafi um, kindly recruited to the project. So I'm thankful for them joining us today. So as you might remember, we have been working towards this data release paper, and now it contains the methods in great depth. We're almost done with writing this up, and now we are sort of focusing on the SLAP2 part. But for the Mesoscope and the NeuroPixel, it's mostly there. And we discussed in the last few weeks a number of figures. I'm just going to drag down to those. Where is the link? Here. Okay. Um, right. So you might remember we sort of talk about having figures that are organized by modality. So let's say 2-photon, NeuroPixels, and SLAP2 as a column. And the first few figure are about explaining how the data was acquired. And then we're planning to have a set of figures about unit quality. So for each modality, we have different process to extract neurons or compartments of neurons. So individual neurons for electrophysiology. So will be units. For mesoscope, we typically we call them a region of interest because we don't always know exactly what's behind the region of interest, and so an analysis that will be helpful for the manuscript will be to sort of characterize the properties of this ROI for the mesoscope, and then later on we could address the SLAP2 validation, which varies subneuronal compartments of different kinds. And so I was hoping we have a focused discussion on the mesoscope validation today. And we were working in the background on receptor field, which is the next figure, but I feel like we could focus on the figure 4 mesoscope work. And this is open for any feedback. I think what would be ideal is if we finish the meeting with a list of things that we think is important to add to this manuscript to validate the region of interest in the mesoscope, and then think about who is doing what towards that goal at the end of the meeting. Does that make sense? Any questions or? Okay. So in terms of data, we now have a large number of both raw files on AWS. So you have access to the entire Mesoscope corpus. There's a few more experiments that are being done, but it will be done this month and package and share. But probably the 3/4 of the data you have access. And then we have also share the NWB files for this session, so you already have access to packaged data with ROI in it by a process involving a variant of Suite 2P. It's a leading segmentation algorithm. And in the NWB, there are traces that are extracted for each individual unit. So what would be helpful in my mind is not necessarily debugging the segmentation, but understanding its output. And so I think there are multiple levels. One is the quality of the ROI within one session, then the quality of the traces, and then the alignment of the ROI across session or some statistics across more than one experiment. And so I wonder if some people have thoughts there, what would be helpful to start. Probably a discussion about the quality of the ROI, region of interest. Have anyone in the group done some of these before? Do you have opinion on the best way to do this? And I could lead it further, but I just want to keep it open. So in the mesoscope, what we will expect is ROI that define somas. There might be some ROI that are in dendrites by mistakes, and so I think there's a classifier that runs. So yep.
Do you have my voice? Sorry for interruption if I made it. Okay, actually, before starting of this session, I started a discussion in Git for today's meeting. Let me just share the screen, and I have some okay, screen. Do you have my screen or?
I can't see your screen. Maybe I'm looking in the wrong place, but.
Oh, okay. Yeah, there is a problem. I think there is a problem with the browser not letting me to share my screen. By the way, if you have access to GitHub, I have a PDF of some suggestions.
Is that in the discussion?
If you, yes, discussion, question and answers part. Yes.
This one?
In Q&A. I didn't put it. It is in the Q&A for. Yeah, the latest one. Yeah, that's today's one. Yeah. Yeah, if you download the first quality control report PDF, the first. Thank you so much. Sorry for.
Let me share the. Oh, you already did a fair amount of work. Let me share the right file. Okay.
Yeah, actually, as you discussed it in the pipeline objectives, I mentioned some of them, what I was thinking of based on the experience that we were doing in the lab. So this is, in the objective ones, in the bullet points, the ones that I've did. Yeah.
This one?
Yes, yeah. Unit array per imaging plane, we have 4 different planes of imaging from different session. Soma dendrite classification quality, as you mentioned, we have imaging from Soma dendrite. Array size and mass plausibilities, then the quality in the traces of fluorescent data that we extracted from each array. So these are the analysis, and the last point is my future idea, cross-session stability. I just did it in just single session. I just wanted to discuss it. If we prove it, we can develop it in more better analysis, better figures, and depth-wise analysis.
Okay. So do you have analysis in this report on the files, or what is it?
Yeah, I explained the ideas here.
Yeah.
In the quality control figures, if you scroll up, there was a quality control. Yes, that's one you pass.
Ali, question. Are
Yes.
they similar to how in our lab we are doing quality control, or you?
Yes, yeah. I just did so much surface analysis to this data. For instance, drift of the DFF data to see that do we have any drift in the data. how the data, DFF data distributed around the mean average. Yet 0.6 is quality control
But
figures.
to ask my
I
question
mean,
again,
yes,
I don't think you
yeah.
answered my question. I'm saying we have a pipeline for quality control in our lab. Is it similar
Yes.
to that, or are you proposing something?
Part of it is similar, not all of it. Part of it. It is
Okay.
more preliminary. These ones, it
The
is a
other
little bit
difference,
preliminary.
Jerome, I guess, is that in my lab we're using Suite 2P, so all the quality control is based on the parameters that we get from Suite 2P, or most of it. How is it for the Allen data? The segmentation happens by which software?
Are you asking Ali or are you asking me personally?
I'm asking you, Jerome. You.
Oh, so we have QC files that are generated in each assets that are generated at the time of the processing, and so it's in the raw data. I don't think it's packaged in the NWB, but there's a little file with each session that store metrics that we calculated. What we have not done, what we have not done for this project is sort of look at the distribution of those metrics.
Yeah.
You know,
And then
to
just one other question.
validate the, yeah. But
What
I
is the
think what Ali is
segmentation?
showing is it looks like another set of really valuable quantification
Right.
that we might decide to select and display
Exactly.
on the
Yeah.
figures.
And then what is the segmentation that you guys are using? What method are you guys using?
I think it's currently a variant of Suite 2P.
Okay. All right.
Actually, we are using cell pose.
Yeah. Which is
Finding arrays.
yeah, Suite 2P runs cell pose within
Exactly.
yeah.
Right,
Yeah,
they
Suite 2P.
can keep going. I just wanted to have a little bit of background information. Yeah.
I think this table is the best thing that we can just have a short discussion on that. Then we can look at some sample figures, discuss more how to develop it, the next output. So these
Okay.
are the output of this pipeline. The one is really basic. For each of the planes that we are recording, we have RI number of counts, how many of them we have in different planes. The second one is SOMA probability. I'm looking at the uh, SOMA and gain rate to see that how much of the RIs recorded are most probably from SOMA or from gain rate. The third one is RI area pixels. I'm looking at the area of the mass that we found with SU2P to see that how the size of each RIs that we have and how they distributed. If there would be really a small fragment that we found that, most probably they are noise. So this
Question,
is
Ali, for the SOMA, then so is that you're looking at the shape there? What is it that you do there? It's like how elongated?
Yeah, for that. Actually, I'm looking at the not exactly the mass. I'm looking somehow mass here.
Okay, so but
I'm
wait
looking
here,
at
wait
if
for a
they
second.
yes.
So in our lab, we need that because we do image dendrite. Do we need it for the Allen data, Jerome? Because I don't think we have the dendrite thing at the Allen because everything is pretty much focused on cortical somas. We have it because with the cerebellum, we can have in the same place both the Purkinje cell dendrites but somas, but I'm not sure that's something we need for the Allen.
Well, I mean, we are, I think for the mesoscope data, we are focusing on somas. That's true. For the SLAP 2, we're recording dendrites, but here with mesoscope. I think it's important that we are, I mean, any labeling will not be perfect. And so there'll be cases where you are segmenting something that you think is a soma, but is not. That could be an artifact. That could be, so I feel like some metrics of quality is helpful. Yeah, but we, as you said, we're for this mesoscope data, we're not aiming to record dendrites. And so, yeah.
Okay.
Yeah, I'm looking at the circular the mass.
And then do you have
how
metrics
long as
for
they are
on the functional
yes,
side?
yeah, it is giving a probability, not it is giving a probability label, not I didn't do it 0-1 binary. It is giving how probable it is SOMA.
And how do you
Actually,
compute
I
that?
have, yeah, if you want, let's discuss this, then I would show the compute, how I computed each metric. So
Oh,
we
okay.
can also discuss the metric. Then I have a standard deviation for DFF data and have variable for each of the ROIs. I'm just looking at the standard deviation of them. Then the other one is DFF robust range 5. I'm looking at the 5% to 95% percentile of the DFF data. Then if I'm looking at the frequency of the data and also the drift rate of the data. This
The
last one is the
frequency,
drift.
you find it on the Oasis output?
I'm looking at the, by frequency, I mean that I'm looking at active neurons and deactive neurons. I should have mentioned it more clearly. I'm looking at the activation of the neuron, how they are active across the session. By frequency, I mean like that, not exactly the frequency of recording. That one. The better name would be activation status for them. In the part 7, I explain each of the analysis algorithm for each of the figures. So for instance, it is the count for number of the ROIs for each plane. It is simple. We are just counting the numbers.
Okay, Adi, 1 second, scroll up, please. I want to see for the functional side one more time. So you're looking at the variability. So that's the standard deviation, right? So you basically want to see that your distribution has a good skewness because those are then the spikes, right?
Yes.
Then you're looking at the how large your the amplitude is, then missing data drifting over this. And tell me again, how you compute the by events you meant the Oasis output.
No, I didn't use any Oasis output.
So what do you mean by how frequent? So you just turn each transient to a binary, so 0, 1, 0.
Yes, yeah. Just
So
I'm looking
if
at
it
the how
passes the threshold,
fluctuating. Yes, yeah.
if it passes
How
the
good
threshold, you call it a
fluctuate.
spike.
Yeah, exactly.
Okay. Okay, I also wrote now to another student in my lab, Jerome, who has done a lot of quality control to get the metrics that he used, and we can kind of see how much of that. But I think, Adi,
Yeah,
you're
we have some metrics like that, but are computed in QCJSON, but is in the assets. You know, like it's a similar idea.
Yeah.
But that's
Yeah,
good.
for the drift rate, I just look at the first average, I mean midpoint, median of the first part of the DFF data, and I compared it with the median of the late part of DFF data during the session to see that how they are drifted. If you have drift, meaning that the median should, yeah. Then I have this analysis algorithm for each figures. I explain how I computed each of them. The question was about the probability one, so my probability distribution, this one.
Can you
So
explain it? I think Nicholas wanted to know how it works.
yeah, just let me have the analysis for myself here to my PC. So mathematical.
It's somewhere else.
Yeah, I'm looking at the better explanation. I had it. Okay. The idea of array quality, I have plane, I have index of that. Then I have a classifier to give me a probability. Then I'm looking for some there.
I mean, are you using the ROI? Because we have a classifier that is trained
Yes.
already, and so you're probably reading the output of it. Is that right?
Yes, yeah. Actually, I had the. So based on the data set, we have that. So I'm looking at the parameters that I'm reading for that. A part of that parameter is a Boolean value that I'm reading from that
right.
sum of probability and is dendrite. Yeah, dendrite
Makes
probability.
sense.
These are the data sets itself.
So are you saying that there's already a classifier trained,
Uh,
and there's a confidence value associated with each, or there's a probability associated with each label, and that classifier was trained not by you, but by Jerome and his team?
yes, in
It
NWB file we have.
was trained by David Fang team. It's using ROI Cat. There's a GitHub repository that I think should be listed in the method section, but we can document it further. But yeah, we train a classifier to distinguish soma from other things using contrastive learning.
So in NWB file we have it already. It is a Boolean now. Yeah.
I think I also have a question about your measurement of drift.
Mhm.
I wonder if there's a better method of analyzing drift
rate,
than simply
yes.
taking the last 10% or the last 10% or first 10% of the data and computing the difference between them.
Yeah, definitely we have it. We have so much better algorithms. This is just a start point for me. I just wanted to look at that. If we are getting good results from it, not obvious differences, okay. If we have obvious differences, definitely it would be better idea to go a higher level of analysis. When the basic analysis giving us what we desire, so it's good. We are good with that. Everybody accepting the simple mathematics. This is my idea. If you suggest, definitely I'm here for learning and things.
I'm curious if anyone has any ideas for what could be a better method.
So we have in the QC files, there's a measure of so with each data set, we require a stack. For each plane, we have a stack, a plus minus 50 micron stack, and there is a metric that is calculated that find at the onset and the end of the session the best matching plane in the stack to measure the Z drift over the course of the session. That should also be in one of the QC JSON that is in the folder. We can document that. I think that would be helpful. I think there might be several ways a cell could drift, right? You could have Z drift. There could be bleaching. You could have you could also the cell could be being damaged, so you could see the sort of calcium event just shrink, um, or the plane just lose out because of motion artifact. I mean, there are many different ways we could lose a cell. That's right. And so probably it's not going to be a one answer fits all situation, but I think Ali's point is right about some metrics are useful. Does that answer your question, Nicolas?
Yes. I'm wondering, so there's Z drift, there's bleaching, there's motion artifacts.
Mhm.
Do you think it's possible to, do you think it would be helpful to create a figure to distinguish these sources of drift? Is that at all
I mean, that
possible?
would be helpful. I think the purpose of the data release paper is, well, first it's for all of you to get familiar with the data itself, like, and then trust the output, and be knowledgeable about any potential issues that we have not detected. And then the other is to build trust beyond this group, for sort of having more people interested in the data. So anything that you any figures that convey those things is helpful. So I just it doesn't need to be in the main set of figures. It could be a supplemental, but I think this could be helpful. Yeah.
So how exactly could a motion artifact contribute toward drift in the signal?
Well, so if you have uncorrected motion artifact, the intensity could just steer away because yes, please, Ali.
Sorry for interruption. For instance, Selcos define this as our ROI. So it is looking at the pixel intensity changes during the session in this point. Then all of a sudden, the subject switch the place of the head and mesoscope is there recording. So instead of looking at here, it is looking at here. So the algorithm find this segment as ROI, but the actual ROI came here because of the motion drift. So it is after a while, we are recording nothing. We are just observing nothing here. This is the idea.
But wouldn't registration correct for motion artifacts? That is, if you're registering each frame, then
so in principle, we run a motion correction algorithm on this data, and so it's corrected. But if, for instance, if you have an experiment with a little more drift than typical, the z-plane is off, and then when the algorithm is trying to pattern match a reference image to the current image, it could suddenly have a little more measurement error at any given frame and make mistake into what the XY offset needs to be applied. And so then you end up with in a situation where the registration is not perfect. And then you just have to be aware that these sources of noise could be impacting your signal. And it's not the dominant issue with this data, but it certainly could be the case that there was one plane, like, because you have 8 plane per session, and some of these planes are deep, some are superficial, and so the quality of the motion correction could be different depending on the depth of the recording. And we just have to be transparent and knowledgeable about it.
I see. So a good figure that, say, looks at motion artifacts and its impact on the data would probably look at that registration quality and how it varies over the course of the session.
Over the course of the session, and I would totally expect that the deeper planes have worse motion correction than the more superficial plane. In some session, not all, but it's like in the mice, for instance, the mice that runs the most could have more of those artifacts. We just have to be aware of it. That's part of the process.
Yeah, I would imagine you would expect more motion artifacts, more registration errors when the mouse is moving.
That's right.
Yeah.
And so I think some figures that highlight those aspects could be helpful. Because in the text, we could describe, okay, we see that in typical motion amount is that amount of microns is corrected. We notice that it's larger in deeper planes, and so that's likely a measurement error, and so you have to be aware that of those particular caveats. Okay.
And you mentioned damage as well. That can also be a source of drift.
So we in the method section, there's a table that showed the power laser that was used for the different depth, and we iterated this table over the years to avoid any potential damage by the laser. I think the team is really careful at checking those one by one, but yeah, I think it's important that this is also sort of checked numerically across the whole data set so that we are confident about it. But yeah, I think
How
if there is
do you
damage,
check
we
it?
would exclude. But yeah,
So are you stating that if the laser is really powerful, the cell will be damaged? How will you how could one assess that damage in the ROIs? And how can
I
one
wonder if
distinguish that from,
yeah. I
say,
don't think it. This is. I don't think this should be in this status. But I wonder if Sheila can want to speak to it. Can you hear me, Sheila?
Yeah, I'm here.
Uh,
So could you
can
repeat the
can,
question?
can you explain to Nicholas how your team is checking for laser damage on all of the assets?
Oh, okay. So the first thing that we really just typically do is make sure that we maintain a safe laser power, like Jerome mentioned. So we do a weekly power check to check like what is the 100% of the laser. And then from there, we can extrapolate the different percentages that we use per depth per session. Each session is also goes through like a quality assurance test. So we look at the depth images from each plane that we're collecting from. We have a view of the time series. We also have a view of the surface vasculature. So a lot of these are like sessions that we acquire over multiple days. So we're able to look at how the brain looks, how the cells look for each of those sessions at any time after the experiment. It's pretty obvious when there's brain damage. You kind of see a cell just get brighter. The whole region of interest also has like kind of major morphological changes. So it stands out pretty easily when there's brain damage. I would say that we flag for that as soon as we see it. The animal is typically removed from the final data set, so you wouldn't see it if it, is removed for brain health issues. Yeah, does that answer your question?
yes, so you said you would see major morphological differences, and this would include changes in the vasculature and
Yep, the vasculature, the placement of the cells around it, like the cells that we report from, you sometimes will just not see them after major changes from laser damage. So I think it's pretty obvious.
And so,
yeah.
oh, I'm sorry.
Please, Nikos, ask your question.
Oh.
That's the point of this meeting.
And so in the data set that we have, did you remove say experimental sessions where the cells were damaged?
I mean, yeah, we typically exclude the animal altogether. So if the data set is not complete, it shouldn't go into the packaging at all. Yeah, we don't have a lot of instances of like brain health issues from imaging. And so if we notice them, they're just removed from the workflow altogether and then replaced with a different animal. I think that's the safest way to do it.
So I'm wondering if since it seems like morphological differences are the main indicator of damage, if it would be helpful to have some kind of metric that evaluates for those morphological differences in the data sets to give some metric of damage and say its impact on drift.
Yeah, so typically when we see these changes, I think it's not like it makes this data set not really a good fit for the analysis. Like obviously we're seeing changes in the morphology that are not necessarily related to the stimulus and that including. So I don't think it's a good idea to just continue with mice anyway that have obvious brain health issues or continuing through the pipeline. So I don't know if we necessarily need to like have something in place for the data, but just saying that like we screen out the possible issues with the mice and they're excluded from this study.
Okay, that makes sense.
I would say that among the potential effects of, experimental effects on the analysis, I think damage is probably the least concern I have because it's been checked carefully by the team. I think potential motion artifacts or drift is more common. And we also screen for these, but you guys being aware that a brain is particularly stable and over is a little less stable is probably important because that would impact, your analysis in one way or another. Yeah, but we could develop metrics to look for a morphology stability if you're curious about it, Nicholas. Like maybe like an SSIM between the beginning and the end, just to check that the overall tissue looks the same over the course of the session. That could be done. Okay.
Okay.
Hali, did you have plots you wanted to share? Or?
Yeah, I have the figures. I also attached I just did the analysis on just single session, as you mentioned it in the OpenScope website. There is a session, NWB session, that's suggested to start an analysis. I use the same session. I also attached the results in the GitHub discussion, one by one.
Let me see. Oh, so let me try to share that. And so you were planning to sort of run this across all session, is that right? Or
yeah, my plan was just look at it for a few sessions. If things looks natural, we can go more deeper analysis and cross-session analysis. This was my idea, cross-session quality analysis. How this analysis,
I see.
how the stability across the session.
I see.
Ali, question, Ali. So these are the metrics. Then did you try to apply a threshold and then get the good ROIs and bad ones and look at some average traces so we see how it in effect it worked?
For now, I didn't do any alignment DFS.
So you're just visualizing
averages,
the distributions for each of them.
Yes,
yeah,
I'm just looking at the
So then that's what you would
so
want.
basic analysis. Yeah, I just started with some basic.
So Davis, do you want to share your? Can you speak or?
Yeah, I can speak. I just didn't want to interrupt anyone.
No, please share your point.
Yeah, I was just wondering if we could get kind of like a high level thing of like what the main sort of QC concerns are, things that you think, or like what, like you mentioned that you have the motion correction, that you use like ROI cat, which I think is not something that we use. I'm just kind of interested in like comparing what our lab's pipeline is currently and better understanding, sort of what you guys are using both to improve our pipeline, but also to see like what the main like goals we should have in mind are for the QC based on how you guys have what you guys have already done, basically.
That's right.
Yeah.
So one thing that I could do with the team is write down a little more documentation on this in the manuscript. I think it seems like it's lacking clearly. At a high level, our QC system check for so we have the things on the rig that are done, as Shida said, operators check during the operation if there are issues of animal stress, issues of photo damage. All of those things are detected as soon as possible by the person watching the experiment. And then once the data is acquired, it gets uploaded on our system on CodeOcean, and then a set of metrics, probably 40 to 50 metrics, are calculated. And we have a number of plots that the people acquiring the data also review like a few days later. And if some of these go out of specs, we would fail the experiment and then in some cases actually repeat it to make sure that we have a data that fits the specs. These specs are, so the biggest occurrence of issues is Z-drift. About, I think, 20% of the planes have those issues. Z-drift could occur, because this is multi-plane data, you might have, as I said, planes that are more stable than others. And so you have to be aware of that. Some planes that we have uploaded might have more Z-drift than we'll like, but we share it because in the experiment, the majority of the planes are fine. And so that's one aspect. There is also stability of the trace. So, Ali mentioned that over the course of a session, you see a decrease in the intensity of that neuron. And so if it goes too low, we flag this as potentially a problem. Because that could be the laser is going down, or the water immersion, or the immersion fluid of the objective is sort of leaking out. So this could happen. And so those are, I think, drift and Z-drift and stability of the recording throughout the sessions, as well as animal stress, are the leading issues that we flag. And then we do not currently check on, the ROI individually, like, which ones are, this is done by a classifier down the line that select the ones that are SOMA and are not. Does that answer your question, or?
Yeah, that answers it. And also too, I'm guessing you've written this. This is in the data release draft.
Yeah,
I
think if
information you just said is already in there.
We probably could have had a little more, I suspect, given you guys' feedback. But in addition, I think, in addition to the one we have, I think it's perfectly fine and probably better if you also contribute your own evaluation of the data, because the more the better, in my opinion. And so what Ali proposed today makes sense to me. But we'll have to make it in a way that the code can be rerun by anyone so that they could recreate those plots. Yeah.
Okay, thank you.
Okay. How do we proceed? And it feels like maybe should we sort of decide on the different main buckets and then split up a little bit what anyone wants to look at in more depth, or does that make sense?
Yeah, that would be the best, I think.
So
yeah, I think perhaps we can make a Google Colab notebook that just perhaps we can do some work at the front end and create a Google Colab notebook that can load individual sessions, but also load multiple sessions, so that everyone can generate figures easily
that's right.
using that notebook. Because I imagine that, yeah, some time will need to be taken to create code that can load multiple sessions at a time, so that can easily be done. So I could take care of that and put that notebook into the meetings GitHub, so that everyone can just kind of focus on the QC analysis and generating these figures.
Actually, as I did these analysis, I need to have clear pipeline. I also shared one. If you want, you can use the same one or improve it to whatever you want.
Do you have code for loading multiple sessions in your notebook?
Yeah.
Okay.
Not just single session. I started with single session, but as you can define a list of the sessions, and it is going to load it in a for loop and save it in a dictionary for each session.
Ali, are you loading from AWS or the NWB files?
NWB files. I use NW, and I'm not downloading them. I'm just directly loading them. it has this.
I think it will be.
Yeah.
Yeah, I think there are two sets of analysis.
Mhm.
One is from the NWB because we also have files in the AWS bucket that could be helpful because there you have access to the raw movies, right? And so
Mhm.
you could extract subset of
Yes.
frames. And so that might be helpful. In addition, yeah, on AWS, you have a mouse and all of the session underneath, so it might be sort of practical to do cross mouse analysis directly from AWS. So I just feel like there, yeah, I think Nicolas could contribute there as well.
Yeah, perhaps so are you using a Jupyter notebook or anything or
Actually,
Google Colab?
I use Jupyter Notebook I have that snippet to load the data in Jupyter Notebook, but the main pipeline for me is just directly you put the code and it is going to give you the figures. With the Python, load this main py, and it is going to load the session, whatever you want. I would share the code, it is well documented. I started we are going to all work on that. I tried my best to do the best documentation I can on the code. We can discuss it more in person.
Yeah,
Okay.
if you can share your notebook, that would be good.
Could we discuss this here? So I know you made the list, Ali, but I feel like signal to noise. I think you could look at ROI extraction quality, event extraction quality. I think this is our two different avenue. Self stability.
By event
Process.
extraction, you're talking about the inferred spikes?
That's
Or
right. How I mean, in the sense that understanding the firing rate,
So is
the SNR of events,
is the
like
plan to
those kind of things,
do the
right?
analysis on delta F over F or on the inferred spikes?
Well, I think we should do both, right?
Yes, I think we need both because we keep looking at both, and a lot of the like ramping, like those go away after we do inferred spikes. So
Signal quality, so it will be a stability, baseline. What else?
but generally, yeah, I don't know how we're going well, for event extraction, one idea would be to look at the calcium kernel. That's something that we do also. That for each neuron, the calcium kernel can get the average, see how it looks like. Does it look like what you expected? But normally a good delta F over F quality will give you a good event extraction.
As motion correction.
You can also add the calcium kernel for the event extraction quality. Oh, you put it in delta F over F. Yeah, I will put that in event extraction.
Well, so the reason I'm writing this up is I think different subgroup of people could focus on different bullets here, right?
Yeah, I'm just saying the calcium kernel should be moved to event extraction, not to DFA. Because that we get from the.
That's right. And I think, Ali, you already touched a number of these, right?
Yes.
But I wonder
Jerome,
if...
why do you prefer it this way instead of dividing the sessions across people?
Well, because why would you want everyone to look at a subset of session? Because then you're going to have different metrics across different session. Whereas if someone is working on a particular problems, he can design the code to answer and understand the distribution.
The code will be a single one that everyone will be using, but no, that either way is fine. I mean, because all of these collectively also perhaps inform the quality of the session. So in that sense, it might be good for one person to evaluate all of them. I thought we're going to do it like session, like one person says like, I'm going to take care of these. The other one
No,
and then each
I
of them.
I feel like it might be easier if we write a set of share notebooks to go across, like Ali and Nicola suggested to do, like tools to study one or many, and then have people write code to look at the different things across all of the session. So it'll be all the things that are listed here.
Okay.
Does that make sense?
Yeah, I mean, both ways work. Like, I guess whether we do one person per metric or one person per session, I don't have a strong opinion for that. I guess each of them comes with its own advantage.
I mean, because some of you are more experienced than others, I think, and so I would advocate for people to take a bite depending on their experience, and then everyone can bring everyone up to the same level. But I feel like these, for instance, is an isolated problem, like it's a separate thing. These is a separate thing. Yeah, this is probably related. So I feel like we might have three groups, and then this is tools across. You see what I mean? Like if someone can build the tools to extract metrics across, and then someone could focus on cell matching and things across session, and then we'll have motion correction, and then these are together.
Well,
Would that make
there's
sense?
also ROI extraction quality at the top. By that, I guess you mean the geometry of the ROI?
Yeah, these to me are related. These three things.
Yeah.
Do you agree?
Yeah.
I'm just trying to organize this. And this is group 2, and I think this might be group 3.
It's troop at this point.
Yeah. Does that make sense? What do you think?
So
Well,
I have a question. So are you proposing that what's the value in, say, looking at an individual session at a time versus multiple sessions at a time?
Or you design your metric on one, right? And then you apply it. It's like you would want, like if you're looking at a single ROI classification, it's probably more valuable to do it at one session first, and then you design your metric, and then you scale up to all of the 20 or more planes. That's how I would do it. Because I think this is what Ali was starting to do. Just write it for one and then scale up.
Just another point that we were discussing, I noticed we can develop our analysis for single session into the planes as we are different depths of recording. We can also add this to look at the quality across all of the planes, then separate it to different planes. Maybe some of them in the future going to use a data set to compare between the depths of the recordings, to see that how this hierarchy connected to each other, how information flows between these. I mean, different planes that we are recording in a single session. So I think that would be
you're already
even
into analysis, right?
yes, yeah. For now, I didn't separate it for different planes. I just look at the counts of the recordings for different planes. For instance, for drift, I look all of them pooled together. But it would be better idea to evaluate the quality across the depths for each of the sessions.
Yeah.
So you're proposing just like have these metrics for each plane separately for an individual session.
Yeah, for individual session.
And I suppose then you could look at for across multiple sessions, what are, say, these metrics for any given plane.
Yeah, also we can do that because one of my own concern is how these information flows between different planes of different neurons. So maybe someone in the future using this data set for this purpose. And if you have lower quality in more depth one, the deepest one. So they can also take it to their analysis, and they have more robust analysis on that. This is how I was thinking. So for instance, motion correction, if we have a motion and recording in the deepest one is going to affect it the more from the motion. So we should also consider that in our analysis.
Yeah. How do you guys prefer to tackle those groups of analysis? Do you want to tackle it together as one group, or you want to split? I mean, I'm just curious. Maybe you want to keep it together? No opinion?
I think making a Jupyter notebook that can be used to easily load the data and look at different planes
That's right.
would be helpful for everyone. As I feel like I see, it seems like focusing on individual sessions at the moment and coming up with some reliable metrics that are convincing to everyone maybe should be the initial priority, and then later we scale up after we trust these metrics and are convinced by them. And I suppose whether or not we're convinced by them would depend on discussions on GitHub in the discussion sections, where maybe some of us can post figures and others can give feedback, and then we can decide, okay, these metrics seem reliable. Let's scale up across sessions.
That makes sense.
That's my idea.
Yeah, that makes sense. So you're proposing that to coordinate the tools to look at one session, I think building upon what Ali started, and then have everybody review those tools so that they work well, and then add metrics depending on the needs that are listed here, and then scale up. So you want to do all of them
Yes,
at
to
once,
multiple
essentially.
sessions.
Yeah. Okay. And who is going to take the lead in sharing those tools? Is it you and Ali?
Actually, I can share my code. So I think from the third party looking at my code, it would be a better idea. You can
Yep.
give more ideas on the matrix, have analysis done. We can develop on that, or even come up with new thing. Problem.
Yep.
With that.
Yep.
And after first stage of have load data, have the metrics, we can come up to groups, develop each parts individually, more better figures, more high quality figures, cross session analysis. We can develop codes by that. In the first stage, I think, starting all together, then separating 3 groups. That would be
I
the
think
best.
at the very least, in either in GitHub or some discussion section, there should be some kind of checklist, I suppose, of like what we want to look at. I mean, I think motion artifacts, I don't think Ali I saw anything to do with motion artifacts. I think having a GitHub poster, however we choose to organize, but we have a checklist. Okay, this is what we want to look at for an individual session. These are the metrics we want to look at for individual sessions. Maybe we'll just take a sample of sessions, like not just one session, but a sample before scaling up to all the sessions. And
Okay.
and that would be a good way to keep things organized.
That's right.
So that nobody's repeating each other's work.
That makes sense to me. So let's start a discussion thread on the forum and go from there. Because I think Ali wrote it, some of it, but maybe as a post instead of a PDF, so that everybody can weigh in.
Sure.
And Farzana, you have multiple people in your group that wants to contribute there, or how do you envision this?
Yeah, so that's how I saw it, that as I said, because they all have other projects in the lab, but at the same time you wrote me and I wanted to make sure we can help with this as much as possible. So I thought I put multiple people so they can divide their load. All of them are also involved with the analysis of 2P data in my lab. So they all have some familiarity at different levels. Some got involved later, some earlier, but it's also like they have done similar analysis for the lab too. So then that can help.
Yeah. So I will check the method section to make sure that all of our methods are properly documented for QC. And then we can start from that and what Ali did into the discussion forum, and then add anything that we think is missing. And then, yeah.
To summarize all of this, making sure I didn't miss anything. So right now, from what I understood, Ali and Nick are going to kind of put together a sort of notebook for all of these metrics. And then different students will take, each of them will take some metric and run across sessions. That's the idea?
That's right. I think we should.
I would say
probably. Yeah, please, Nico.
I would say rather we would make a notebook that just includes code for loading sessions, individual sessions or multiple sessions, or a sample of sessions where we say we compare good sessions versus bad sessions. As I believe Davis mentioned in the chat. And we just make that notebook so that anybody can get into it and look at, say, motion artifacts or system integrity or temporal synchronization or whatever other artifacts or things they want to look at. But the notebook, I think, shouldn't include any analysis. It should just be purely for loading the data, in my opinion.
Yeah, my suggestion was in the first, I would share the analysis code I have. We are going to develop these loading sessions so everybody can jump into the analysis, not deal with how to load sessions, different sessions. So they start quality control. We already did that. Then meanwhile, based on their interests, we are going to separate. I think this would be the best thing. So for every analysis, we know who is responsible. So we can discuss for that analysis, have to develop figures. What are the main figures? This was my idea. Otherwise, if we develop the analysis, for me, as I'm doing another project, that would be more workload because that developing the analysis matrix takes some time. So I think that initial point, we are start from the same point, but in somewhere we should analyze, everybody should take the responsibility of their own analysis and develop their own metrics. So based on the metric, they are going to generate their desired figures and we are going to share that. This was my initial idea. Otherwise, you can suggest and we can do whatever you suggest.
Yeah, I think these three groups to me make sense because and probably in this order. Like someone needs to tackle motion correction stability because that's an important caveat. And then you have to understand the ROI and the extraction of traces. And then this is about stability across session. These are three different P's that anyone could tackle independently of another.
If Jerome were going to add one more here, I would take the event extraction as a separate one. It doesn't have to be, but that's also a separate algorithm than the So
that might be true if you want, yeah.
I mean, depends on whether we want to have fewer or more groups, but I mean, that's where we can potentially do that separately.
That's right. But I feel like anyone should think which one is more interesting to them. Probably. But I would, yeah. So let's go in the discussion forum and continue there.
Yes.
Yeah. Yes.
Jerome, please scroll up a bit. Scroll up so I can take a screenshot of this and discuss it with my students to see who is more exactly.
You want to see this, or?
Yeah, just an easy way. Yeah, that's good. I took it. It's fine. Thanks.
Next week, we will discuss, I think there's a presentation from Sarah Rudiger Group. Just want to highlight before we split. The 19. So there's 2 students that are going to talk about SLAP2 analysis on the pilot data. So the first session will be full with that presentation. Okay, well, thanks so much for all you've done, Ali, already, and I'm looking forward to see all you guys come up with
Thanks
to see in the data.
Thank you so much. Thank you.
Bye.
Goodbye.
See you.
Okay. Goodbye.